In the 1920s, there was a farmer that had moldy hay that he fed to his cows. Well, the cows, some of them died, but others, they had bleeding in their teeth and on their udders when they got milked. And the farmer didn't know what was happening. So he had investigators come in and they found a toxic substance in the mold, and they called it coumadin or warfarin. And they eventually gave this to rats in a higher dose and killed the rats pretty quickly. Well, some of them were familiar with Paracelsus, who had stated that everything can be poisonous, heck, even water can be poisonous. But from some things, you can change that dosage and it can be a remedy. So what we're going to do today is, we're looking at how to use this as a remedy. Hey, Dad, when you have AFib, right? So the blood's pulling in your heart. If we give you some warfarin, it will stop you from having a stroke and from pulmonary embolisms and from deep venous thrombosis. Dad, where are you going? I was just going to use 5 milligrams. Dad, I was only going to give you a very small and therapeutic dose. So I used the example of coumadin or warfarin because it can be monitored how well it's working in the serum. It's called a PTT or INR, and at one range it works very well for people with AFib, and at another range it works very well for people who have had valve replacements. So that can be monitored just as I want to monitor the effect of our chaperone, whether it's 4-PBA or TUDCA or another chaperone, and we want to look at markers in the UPR and get that dosage perfect. CEBP homologous protein, aka CHOP, is a member of the CCAA T enhancer binding protein family of DNA binding transcription factors. Well, what does that mean? There's a sequence. It's called a cat box, and it has that CCAT sequence in it. And it is usually 60 to 100 base pairs upstream of where the initial transcription factor binds to transcribe DNA into RNA. It is a pro-apoptotic transcription factor that is ubiquitously expressed throughout the cell, and you can also find it in the serum. And there's many mouse models that study this particular factor. And in the diabetic mouse model, they have insulin that is being expressed at a high level, and it is causing insulin resistance and inflammation throughout the mouse. But when you have a deleted gene that expresses CHOP, you have reduced oxidative stress, and the beta cells in the pancreas are less stressed, and they produce then a therapeutic amount of insulin. So let's begin looking at some journals that we can look at GRP78 and CHOP in a clinical scenario. The first one here, circulating GRP or BIP, is a marker of metabolic diseases and atherosclerosis. and it brings endoplasmic reticulum stress into the clinical scenario. So they have several objectives in this paper. One is to look at how GRP78 responds to obesity, diabetes, and metabolic syndrome. And the second is to look at nicotinamide, which is a B vitamin, and how it can act as a chaperone to affect these diseases and the level of GRP78, and also look at fenofibrate, whether it affects it. Now, fenofibrate is a medication that you take for increased triglycerides or increased cholesterol. So it'll help you control your fats. So if you look at the obesity here, and these are all looking at GRP78 or BIP, you could be obese and not have a big change. And that's kind of, you could see how a person can hold their weight. If they're big around the middle, that's a key indicator that they are heading toward metabolic syndrome and diabetes. When you look at it with just comparing diabetics with non-diabetics, very significant. Say 9 to 17, very significant difference. Same with metabolic syndrome, which is rated right here. So you can rate metabolic syndrome as, look at things like pre-diabetes. Where is the insulin? Where is the sugar? Where are they carrying their fat? Once they become a diabetic, and then at different stages of diabetes, do they have to use insulin? Are their kidneys shot? Are their organs starting to go? Are they getting atherosclerosis? In this slide, GRP78 is related to plaque. If they have a significant amount of plaque, they're here with a GRP maybe of 24 compared to negligible plaque with a GRP78 of maybe 12. So now we get to look at the GRP78 when treated with fenofibrate and with nicotinamide. Here's our control, and it's close to a 3. Type 2 diabetic, close to a 4.6. Treated with fenofibrate, 4.5, doesn't matter really much. Here it's made a significant difference, and this is treated with 2 grams of nicotinamide a day. And that's why I called it a weak chaperone. It did not bring down BIP to the level of the control or closer to the level of the control. I would have liked to have seen it around here. And that's why I mean by adjusting a dosage of a chaperone to what we see as far as a marker such as GRP78. This journal looks at the circulating levels of GRP and of CHOP in Chinese type 2 diabetic kidney disease patients. And they conclude that patients with type 2 diabetes mellitus showed increasing circulating levels of GRP78 and of CHOP concentrations. Interesting enough, as the kidney disease increased and the glomerulus leaked out albumin, these levels also went down because they leak into the urine. As a nurse, I've seen patients come into the ER with sepsis, and it is pertinent that you get a diagnosis quickly with these patients so they can be treated appropriately. If not, they will die. So when someone comes in with altered mental status and low blood pressure and an increased respiration rate, we want to check for organ failure. And we want to run appropriate labs that could indicate that they have an infection and that they have sepsis. In this paper, they particularly look at GRP78 and CHOPs as a diagnostic tool. And when this particular lab looked at those cutoff points, they plotted it on a chart that has sensitivity for the disease, in this case sepsis, and for being negative to the disease. That's called the specificity, if you can rule out the disease. So these represent the days. So on day 1, it was sensitive. On day 2 is the green one, and it was the most sensitive to be measured and indicate that the patient would have sepsis on day 2. On day 3, it lost its sensitivity and ability to predict that the patient had sepsis. In this graph over here, they look at the sensitivity of GRP78 to predict sepsis. And again, the green line here predicts it is most sensitive to predict that this patient would have sepsis. And that is indicative of day 2. Okay, when we look at other markers, such as interleukin-6 or C-reactive protein or procalcitonin, looks like the procalcitonin is the most predictive. And next would be the purple line, which looks like GRP78 on day 2, along with CHOP on day 2. Pretty good. And another study shows that physical exercise can alleviate ER stress, and that being lowering the GRP78 chaperone in the serum. It is also shown in some studies that fasting can do the same thing. There may be other markers out there like caspase-3 that could be found in a serum. It is increased with apoptosis and antiglomerular basement membrane nephritis. And it can be found right next to CHOP in initiating apoptosis. But I really believe that we can use these as markers, BIP, because it is the main chaperone in the endoplasmic reticulum. It's going to show us a lot. And CHOP because it mediates ER stress-induced apoptosis. And other markers may be more specific for certain diseases and certain symptoms in various patients. Using these markers can help us get a chaperone dose that is not too high as to push BIP down too low, or to have a not strong enough dose so that it doesn't push it down low enough or normalize it. So we want that porridge to be just right. Chaperone. What is porridge anyway? At any rate, like, subscribe, check me out on Collagen and Rare Disease Foundation.com. The next video is going to look at specific numbers with dosing in mice and humans and specific concentrations for chaperones in tissue culture. Okay. I'm hungry. Hey, do we have any porridge? Dad, rat point warfarin is perfectly safe. We just need to take a little bit of blood. Where tried on cows, well, I won't have any left after that. What do you think?